We're moving on now to looking at the immune system in quite a lot of detail. So this should be quite an interesting topic for a lot of people, and it's also pretty relevant, some of the things we're going to talk about, with what's going on at the moment with coronavirus around the world. This is all the information for this topic. Don't get too overwhelmed. There's a lot in here, but we'll spend a bit of time on this. What we're going to look at today is these first two sections. So one is these things called antigens, and then we're also going to look at the pathogens. We'll actually start with pathogens because I think that probably just makes a bit more sense, and then we'll look at antigens. All right, you are going to need to draw a bit of a table in your notes here. So if you want to go through this slide and then do the table afterwards, you can. Really important that you have a definition for what a pathogen is. So a pathogen is basically an agent that causes disease. Okay, and when we go through the examples, you've probably heard of most of these before, but a nice, easy summary, something that causes disease. I think it's worth adding this definition as well of what infection is. We often think about infection as being, when you're sick or you've got a wound and it's infected and it gets all pussy and gross. But basically an infection is when a pathogen has entered a host. So when something that can cause disease has entered the body in some way. And we'll look a bit more at how that happens later on. Okay, these are the pathogens that VCAR say that you need to know. So 6 different types of pathogens. And there's a few key things about them that you need to know. So you'll notice at the top of this table, it kind of describes the pathogen based on its structure. So we have unicellular pathogens. So they are pathogens that are just one cell on their own. We have a few multicellular pathogens. So they're made up of multiple cells. So they're a bit more complex than a unicellular pathogen. And then we also have non-cellular pathogens, or you could say non-living pathogens. And when we go through those, I'll explain what we mean by non-living pathogens. So we'll start over with bacteria. Now, I'll just note now, bacteria and viruses, we look at the most, and you'll do a more detailed drawing of these on the next few slides. So if you want to take this image for your table, you can, or if you want to draw a nice simple diagram in your table, that's absolutely fine. So bacteria are prokaryotic cells. Okay, you looked at this in year 11, so they are simple unicellular cells, and they don't have membrane-bound organelles. They divide through binary fission, and we'll quickly recap that in the next couple of slides as well. And even though they are unicellular, so one cell is a whole bacteria, they tend to, well, because of the way they divide, they clump and kind of live in what we call colonies. So a colony is basically just a bunch of bacteria, the same type of bacteria, kind of living or existing together. And some examples of diseases caused by bacteria are things like tetanus or meningitis. You don't need to know specific diseases related to each type of pathogen, but it's, I'll put in ones that you've probably heard of so you can start to identify them. The other type of unicellular pathogen are called protozoa, or protozoon is a single protozoa. Now these are unicellular, but they are eukaryotic cells, which means they have organelles. And you'll see it's a bit blurry, but in this diagram you can see there's mitochondria, there's Golgi apparatus, there's ER, there's the nucleus. Okay, so they're a more complex cell than a bacteria. They can be free living, so basically just existing on their own and being able to survive on their own, or parasitic. There's a few things in this that we talk about as being parasitic or being parasites. So I've got this little definition down the bottom here. So a parasite is anything that takes its nutrients from their host. Okay, so they can't survive on their own. They need to use nutrients from the host. Okay, so some protozoa are like that, and some of them are free living. An example of a disease caused by protozoa is malaria. And you'll probably associate malaria with mosquitoes. And that is true. Mosquitoes carry the protozoa that cause malaria around. So this is just a handy word to know. A vector is something that carries the diseased organism around. Okay, so for example, a mosquito is a vector for the protozoa that causes malaria. We'll move into our multicellular pathogens. So the first one is fungi, okay, which, mushrooms are a form of fungi, like a much larger form. When we look at fungi that cause disease, we're looking at much smaller organisms. So things like yeast and mold are examples of fungi. They're eukaryotic, so they're complex cells with organelles, but we call them an organism because they're not just one cell. So in this picture, each of these little things is a cell, and those dark dots are the nuclei. Okay, so they are an organism, they're multiple cells. They are ectoparasites. So if we go back down to our parasite definition, ectoparasites live on the surface of their host. And you can see here from the examples of diseases caused by fungi, things like thrush or athlete's foot or ringworm, so often things that affect the skin, because fungi are ectoparasites, they live on the surface, and when we said they get nutrients, the nutrients that they would take would be things like dead skin, and that's why they're often associated with skin conditions. Then we have worms, which are, again, eukaryotic organisms. They're made up of multiple cells, and they actually are, similar to a worm that you would find in the ground, but much, much smaller initially. So they have a mouth, they have intestines, they have an anus. Okay, so when they take nutrients, they will process them and get rid of waste. Worms are endoparasites. Usually, there are some that live more on the skin, but in general, worms are endoparasites, which means they live inside the host. So a very common place for worms to be found are in the intestines. Anyone who has pet dogs or cats, you probably give that animal deworming tablets, and that is to get rid of any worms that might be living inside them that make them sick by stealing their nutrients. Okay, so an example of that is tapeworm. If you're like into horror movies and things like that, jump on Google and look at some parasitic worms. They're pretty gross. Then we move on to our non-cellular pathogens. So we have viruses. Like I said, we'll look at viruses in more detail, but a virus is basically genetic material. So here we're talking about DNA or RNA in a protein capsid, which is a shell. Okay, so they're very, very simple. It's just a bit of DNA or RNA in a protein shell. They need a host to reproduce. So if we come back up to here, how we said they're non-cellular, which means they're non-living, that is because a virus cannot reproduce on its own. It needs to move from host to host, and we'll look at that in a little bit more detail. Examples of this, obviously coronavirus is a virus that causes the COVID disease. HIV is a virus, human immunodeficiency virus, which causes AIDS. Influenza is a virus, Ebola, there's heaps and heaps of different examples. And our last non-cellular pathogen are prions. And you may remember this coming up in your Year 11 SAC. A prion is a protein that hasn't folded properly. So if we remember how a polypeptide chain goes through these changes and folds into a shape, a prion is a protein that folded abnormally, and then it can actually affect the proteins around it and make them fold abnormally. They're only found in mammals, and they affect the proteins that are found in the brain. And an example of this is mad cow disease. Okay, so that was a big thing in the 80s, and it was a disease that affected the brain tissue initially in cows and then in people. Again, you don't need to know these examples, but any of these or different examples of disease could come up in your exam or in a SAC as part of the stimulus material. So if you'd like to pause here and make sure you've got all of these details written in your table, and make sure you've got whether they are unicellular, multicellular, or non-cellular. And then we'll move on in a bit more detail. These are just some electron microscope images. So we have bacteria over here. So you'll see it's got that kind of DNA that inside, and then the outer cell wall. It's got these flagellum, which we'll go through that in a second, but often they have tails so they can move around. Here we have protozoa. Okay, so you can see in there the organelles. It's a bit more complex than the bacteria, and it's got that kind of plasma membrane around it that you can see quite clearly. Here is a prion. Okay, so it's a protein that's not folded the way it should be. Got down here, fungi. Okay, so you can see these individual cells making up the total organism. We have a parasitic worm here, so you can see it's got that worm shape. You can kind of see the cells inside it, and then finally we have a virus. Okay, so we've got some genetic material in here. We've got the shell outside. We've also got these little markers all around it, and we'll talk more about them in a little while. Okay, you should have a more detailed diagram of a bacteria than the one we had in the table. This is important, well, for this topic, but also really important, we come back to this in Unit 4. So it doesn't have to be, a nice 3D one like this, but you do need to have these components. So a bacteria, it's a prokaryote, so it has a cell membrane. And then it also has that extra layer of a cell wall. Okay, so that is really, really important. That cell wall protects bacteria in their environment. Bacteria just have a single circular DNA chromosome. Okay, so where you write chromosome, you might want to just add DNA in there, and it's just one chromosome, not like our cells, which have 46 chromosomes. They also have these little things called plasmids. Now, draw that, label that. You can even add that that is also made up of DNA, but we'll come back to that much later on in Unit 4. But that is an important component of bacteria. They don't have membrane-bound organelles, but they do have ribosomes. Okay, so bacteria need to make proteins, so they still have ribosomes. They have a cytoplasm, so they've obviously got the liquid inside. And then these next two things are found in some bacteria, not all bacteria, but some bacteria have things called pili, which are almost like little hairs on the outside, and some of them have a flagellum, which is like a tail. They might have multiple of those, and they're really to help protect the bacteria or to help it move around. This should be revision from Year 11. But binary fission is the way bacteria divide. Okay, so while our cells go through mitosis, bacteria go through binary fission. It is a type of asexual reproduction. So bacteria do not need a partner to divide. And the process itself is quite simple. We get the DNA doubling, because it's going to need DNA in each of the cells. The cell kind of stretches and elongates, and then it breaks into the two cells, and the new cell walls will form around it. So now you've got two identical copies of that first bacteria. A thing to note, I guess, here is that the way they divide is kind of exponential. So we started with 1, we ended up with 2. If each of these 2 divide, we'll have 4. If each of those 4 divide, we would have 8. Then we would have 16. Then we would have 32. Then we would have 64. Okay, so it would divide in kind of an exponential growth. And that's quite important when we look at how disease can spread. Again, you need to have a more detailed diagram of a virus. Just remember, all viruses look very different. So this is a nice general diagram that you might see of one, but they do all look quite different. So we have in here either our DNA or RNA. Okay, so it's worth putting that in. They'll have some kind of genetic material. Then we've got around it our capsid. Okay, so that is the protein shell. You could even, when you do your diagram, write that that is made out of protein. And then some viruses are just that, just the DNA inside the capsid. A lot of viruses also have what's called an envelope. So that is a protective layer around the whole thing. And then ones that have an envelope would also have what's called envelope proteins around it. The way viruses reproduce is different to bacteria. I think it's worth drawing this diagram and having an understanding. The key point is that they need a host to reproduce. So bacteria can just reproduce on their own. Viruses need to use our cells. So to do that, a virus will first attach to a cell. It will then use the cell's organelles it will release its genetic material. So its DNA will get released into the cell, which you can see here. And then it will use that cell to basically reproduce itself. So it's basically told the nucleus of this cell to start transcribing and producing new copies of itself. And then it will form its new capsids and envelopes, and then usually those viruses will leave the cell and destroy it. Sometimes the cell will not be destroyed, but usually or often the cell is destroyed. Okay, so the important thing here is a virus needs to use host cells in order to reproduce. And one cell might make hundreds of copies of that cell, of that virus. Sorry. I just added this in because it's quite interesting and I thought it was quite cute. This is actually a painting that a biologist recently did of the structure of the coronavirus. So viruses tend to have these really beautiful geometric shapes. And so this was a diagram he did of all the genetic material in here. And then we've got our capsid and our envelope around it. Okay, so we've had a look at the different types of pathogens. What we're going to move on to now is these things called antigens. Okay, so it's very related to pathogens, but this is another key term that you need to know. So antigens are substances that interact with our immune system. When we look at the examples, that should make a bit more sense, but this is just a good definition for you to write down. What antigens do, in a nutshell, is they tell your immune system whether something that is in your body is foreign or whether it's part of you. Okay, and that's really, this is the key thing really to our immune system, is our body's ability to tell which cells inside the body are your own cells and what is coming from outside the body and shouldn't be there. Based on that, the names kind of make sense. We have what are called non-self antigens. These are markers or substances that are found on pathogens or foreign things. Then we have self-antigens, and these are the markers or the substances that are found on your own cells that tell your body, tell your immune system that they should be there. It's part of you. Okay, so if we had a bacteria, we might have these little markers all over it. They are antigens, they are non-self antigens, and they would tell your immune system, I shouldn't be here. If you had one of your own cells, give it a smiley face because it's one of your cells, and they might have different markers that say, yep, don't kill me, I'm part of you. We'll look at these in a bit more detail separately. So we'll start with self-antigens. So self-antigens are the markers that are found on your own cells. Their job is to make sure that your immune system doesn't attack your own cells, because that would be a disaster. You do need to know this term, okay, especially this abbreviation of it. So make sure you write this down. In humans, and when we look at the immune system, we're mainly going to use human examples because we can relate to that quite well. So in humans, we have a type of self-antigen, or two types of self-antigens, called Mhc markers, or major histocompatibility complex markers. Now I know that is a mouthful, but for people that kind of like breaking down words, I'm just going to do that for you. Histo means tissue. So histology is the study of tissue, so body tissue and body cells. And compatibility obviously means that something fits, or it's compatible, or it should be there. Okay, so if you look at that, histocompatibility is basically saying that this tissue is compatible. It's part of me. There are two types of MHC markers. MHC1 markers are found on all of the cells in our body except red blood cells. Red blood cells don't have MHC markers. Then we have MHC2 markers, which are found on certain cells in our immune system. And we'll come back to that. They're really important in terms of our immune response. We'll come back to that. So all cells have MHC1. I'm just going to draw it like that. Some cells, which are immune cells, would have MHC. We'll go immune. They would have MHC1 as well, because all cells have that, but they would have MHC2 also. And they're just little markers on the outside of the cell, like this. This top diagram, people often find a little bit confusing, but we'll break it down. This is the cell that we're looking at here. Again, I'll make it a smiley face. This is one of your cells. This is the self cell. Oh, my eye disappeared. This is weird. A T cell is an example of an immune cell. So this cell would also be in your body, but what we're talking about here is this cell. We don't want the immune system to attack this cell, so it has these little self-antigens that say, don't kill me, I am a self cell. Sometimes the immune system makes mistakes here. So if the immune system mistakes self-antigens and accidentally attacks them, so instead of seeing a cell with a self-antigen as being part of you, it thinks that it's foreign and attacks them. That can cause what we call autoimmune diseases. So this is when your immune system attacks your own cells. And some examples of autoimmune diseases are things like rheumatoid arthritis, where the immune system attacks some of the cells in a person's joints and causes a lot of pain. It can cause psoriasis, so when the immune system attacks a person's own skin cells and causes skin problems. And also multiple sclerosis, or MS, is when the immune system attacks a person's nerves, and that can cause disability. So they're just some, it's worth noting this down, we'll come back to this later, but sometimes problems happen and the body doesn't recognize something as being a self-antigen. Right, non-self-antigens are something we're going to look at a lot when we look at the immune system. So these are the markers that are found on things that originate outside of the host's body. Things is probably a terrible term to use there, but usually what we're talking about is things like cells or pathogens. Non-self-antigens are what trigger an immune response. So when your body identifies antigens that aren't from one of your cells, they will launch an immune response. If we go back to this diagram here, I'm going to put a sad face on this one because it's not a self cell. This is a bacteria, for example, that we don't want in our body. This is your T cell, so one of your immune cells. If that picks up this marker and says, I don't recognize you, you're not part of me, it will launch an immune response to try and get rid of this pathogen. Non-self-antigens are not, they're often proteins, but they can also be things like sugars or genetic material. And they are found on all pathogens. So a really important thing about immunity is that our body can actually recognize things that shouldn't be there. A couple of little things to note, and again, we'll probably come back to these a little bit later. Allergens, so you've probably heard of allergic reactions, or you might even have some yourself. Allergens are antigens that are found on non-pathogens that actually shouldn't be harmful to us. Okay, so remember, pathogens are the things that cause disease. So a non-pathogen might be something that's not part of us, but that doesn't cause disease. So for example, peanuts is a really good example of this. Okay, peanuts are not pathogens. For most people, they don't cause disease. They're quite delicious. But for some people, their body recognizes these markers on the surface of peanuts as a pathogen. And then their body will trigger a really exaggerated immune response. So the body will pick up these markers and launch a really heavy immune response. And that's what's called an allergic reaction. And that can be really, really dangerous for people that have that. Okay, so things like peanuts, a lot of people get it with pollen, so things like hay fever. All of those things are examples of allergens when the body's immune response is a bit overactive. Another really interesting thing here is, remember we talked about our Mhc markers? So they are self-antigens. An important thing to note here is that my self-antigens are going to be different to other people's self-antigens. Okay, some people have the same ones, but often different people will have different self-antigens. So what is self to me wouldn't be self to someone else. And that's really important because if someone else's cells end up in your body, that can trigger an immune response. And a really common example of that is if someone had an organ transplant, or even say a blood transfusion, the person who receives that organ or that blood, their body can reject it. That can actually make them really, really sick because their body is identifying the Mhc markers from the donor's cells as being foreign and launching a response as they would for a pathogen. So this is why if anyone's ever donated blood or things like that, they obviously test what type of blood you have, what type of markers you have, to make sure they don't go into a person that has a different type of marker, because their body will reject that. Right, this is a question from a previous exam that relates to this. So if you want to pause and have a read through and see if you can figure it out, and then come back and we'll go through it. So this is basically saying that there are 3 different types of structures found on cells. They have their own antigens, so they're self-antigens. They have receptors that will enable their cell to identify its own antigens on any other cell, and then receptors that pick up the foreign cells. Basically what the question is asking is which one of these diagrams shows that is a self-antigen for a particular cell. So given that we've got 4 different types of cell, a self-antigen for that cell will have to be unique to that cell. Okay, so for example, the cells in your liver would have different self-antigens to the cells in your hair. Okay, so we're basically trying to find where a unique antigen comes up. So this first one, is this a self-antigen for cell P? So if we look at cell P, it does have that antigen. Is that antigen found in any of the other cells? It's not in cell Q, it's not in cell R, but it is over here in cell S. So that is not unique to cell P. That would not make it a self-antigen. Okay. If you struggled with this question, but now you think you've got it, you can pause here and figure out which one it is. But I'll continue on. So this structure, self-antigen for cell R. So we've got that here. Have we got it anywhere else? I don't think so. So we'll think this is a possible, but we'll eliminate the others first to make sure we're on the right track. Okay, is this a self-antigen for cell Q? So that is found here, but it's also found in cell R. So it's not that, it's not unique to cell Q. And finally, is this a self-antigen for cell S? So it is found here. It's also found on cell Q, so it's not unique. So the only one here that was unique to that cell was B, which was the self-antigen for cell R. Okay, I want you to go into Edrolo and answer the questions. So this is section 9A, and if you're doing the questions in your textbook, they're page 305 to 309. I want everyone to have a go at questions 1 to 10. If you think that is kind of making a lot of sense and you want to test your knowledge a bit further, you're welcome to have a go at questions 11 through to 17 as well. And we'll be continuing on with the immune system next week after we've done our SAP.